we're going to start talking about the papillomaviruses. I haven't really talked about human papillomavirus as yet, and many would probably classify HPV more in an oncology block, because as many of you are probably aware, HPV is a leading cause of cervical cancers, but there are some other clinical syndromes that are associated with HPV infection, and then there's also a lot of asymptomatic cases. So HPV is kind of another one of those viruses that can run the gamut. The papillomavirus family is actually very similar to the polyomavirus family. We have talked about a couple polyomaviruses. Most notably, we talked about JC virus during your brain behavior and cognition block, which in severely immunocompromised patients can lead to progressive multifocal leukoencephalopathy. Remember, JC virus is one of those organisms that kind of we're all infected with probably already, and our brain kind of keeps it under control using a good immune response. When the immune response is compromised, it grows out. Another one is BK virus, which kind of works similarly and is often associated with kidney infections. And like actually the papillomavirus can lead to kidney cancers. So the papillomas and the polyomas actually have some similarities in characteristics and how they function. So like the polyomaviruses, the papillomaviruses are small, non-enveloped organisms. They have an icosahedral capsid and a circular double-stranded DNA genome. There are a ton of HPV viruses. There are more than 150 different types, and less than 90% DNA homology. And the way we get the types is basically if you have less than 90% DNA homology in a specific viral protein, L1, then it's considered a different type of HPV. The different types, so you've got 150 different types. I'm not going to draw 150 dots, but then the types are grouped based on how similar they are to each other. And there are 16 different groups that are named A through P. Okay, so that's just kind of the structure of how this actually works. With the HPVs, the way they function is that they infect susceptible tissue. So most cases, what we're talking about with susceptible tissue is cutaneous or mucosal, which when you think about this for where you might be exposed, that's the sites of how this could work when we're thinking about this as an STI. Cutaneous and mucosal is an excellent opportunity for these viruses to infect. They are highly resistant to inactivation on surfaces, so it's somewhat difficult to get rid of them. All right, so I already went over most of what's on this screen. So they're like the polyomas. They're small, non-enveloped, icosahedral, the double-stranded circular DNA. What I didn't talk about was kind of their life cycle. So part of the reason that the papillomaviruses cause cancer is that they kind of have this double life, okay? In a permissive cell type, they are a lytic organism. So they'll grow, they'll replicate, and then the cell will burst, and that will release the virions. They're able to access non-permissive cells and to kind of start the replicative process in non-permissive cells. And what happens is that then becomes oncogenic. So when we see HPV-induced cervical cancer, or the types of cancer that HPV can cause, like throat cancer, it's because the virus has infiltrated a non-permissive cell type, and that's led to kind of the outgrowth of cancer. So let's think about just kind of the different genes that we might see in an HPV genome. So it's double-stranded, circular DNA genome, and it has two sets of genes. It has an early gene and a late gene. This should be somewhat familiar because the classification isn't that different from herpes virus, right? Where we have very early, and late. So now here we have early and late. There are 7 to 8 early genes, depending on the virus, because remember, there's all these different types, and 2 late structural genes. And remember, the types are based on the structure, the DNA homology of the L1 structural gene. There's an upstream regulatory region that basically controls the sequence for the transcription of these different types of genes. And there are overlapping reading frames to maximize the coding potential of the virus. All of these genes are located on the plus sense, right? So if I'm gonna attempt to draw some circular double-stranded coiled DNA here, okay? Ignore my shoe case. So if this one is your plus sense strand, and this one over here is your negative strand, all of the genes are going to be on this plus sense strand. And they're transcribed from that strand. And then the gene expression is also controlled temporally by the order of the expression. So we're going to express one type of gene and then the next type of gene. All right, let's talk a little bit more in depth about the replication of the human papillomaviruses. So in this case, the virus tries to follow the maturation and replication of the cells that it infects naturally, that it's permissible in. And this actually makes a lot of sense when you think about the oncogenic properties of HPV. Since this virus is trying to grow with the cell type as it changes, you can kind of understand how when it gets into a cell that doesn't follow the normal program that it's expecting, that the cell then would be affected by the presence of the virus, and that could down the line lead to cancer. Okay, so let's talk about what's normal for HPV viral replication. So HPVs want to infect 2 types of cells, namely keratinocytes and epithelial cells. It's going to access the basal cell layer through breaks in the skin. So scratches, abrasions, grooming, anything that would lead to a break in the skin area where you could potentially be contacting the virus, okay? It's going to access the keratinocytes or the epithelial cells at the basal cell layer all the way down here. Okay, oh, I forgot. I'm using white, so you can't see that. All the way down here, all right? Then what's going to happen is that L1, which is, remember, one of those structural proteins, that's going to actually be the viral attachment protein. So go back all the way to the beginning of your M1 year, and we talked about how viral attachment proteins are basically just glycoproteins on the surface of virions that bind to host cell receptors, allowing for receptor-mediated endocytosis into the cell. That's going to initiate the replication event, and it's doing this by binding to heparin proteoglycans and integrin alpha-6 and other receptors. So the main one that you might think of is the integrin alpha-6, but really it's heparin, okay? All right, then we're going to have the activity of the early genes. So the early genes are basically going to tell this cell to grow. Okay, it's going to stimulate cell growth because remember, the virus needs the cell to grow in order for it to grow. They grow together, like an old married couple. So it's going to stimulate host cell growth. And what happens is it facilitates the replication of the viral genome by the host cell DNA polymerase. So the host cell needs to have its DNA polymerase active in order for the virus to be replicated. So that's why it wants that. Now, this is where things start getting tricky. The virus inducing this increase in cell number means that we're going to have a lot more keratinocytes and epithelial cells than we would normally have. Okay, this happens really fast. And that's actually what's going to lead to the production of warts, because basically what warts are is a thickening of the cell. Another term for this is condyloma, or very appropriately, papilloma. These are other terms you might see for the warts that are caused by HPV. All right, now let's get back to our differentiating cell. So as this basal cell down here differentiates the specific nuclear factors, transcription factors, etc., that are expressed at each level of skin development, promote transcription of different viral genes, right? We've got some of those early genes, we've got some of those late genes. The late genes are going to encode structural proteins that will only appear in the terminally differentiated skin layer. So remember, we need that virus to get all the way down to the basal cell layer where it can infect, then it's going to mature with the cell, and then as it becomes a terminally differentiated skin cell, now it has the structural proteins that make the virion. Now remember, we are all dropping dead skin cells all over the place, all over the world right now. You're dropping some dead skin cells. If you have HPV, now these dead skin cells are full of virions that can be shed with the dead skin. Okay, so they're in the terminally differentiated upper layer, and as this infected skin cell matures its way to the surface, the virus matures as well, and when the dead skin cells are shed, the virus is shed as well. Okay, now that's the way it's supposed to work, and we'll talk about what happens in cancer a little bit later. So given that we now understand like the normal rate that papillomas are maturing with skin cells maturing, it's pretty easy to think about how we get this. Close contact is basically your number one way, and that's through sexual intercourse, because obviously we're going to have some shedding of some dead skin cells there. The other way that people don't think of as often is birth, as the baby makes its way through the birth canal, can probably be exposed to some dead skin cells, but really we're talking about sexual intercourse. It is actually the most prevalent sexually transmitted infection in the world. Greater than 20 million people worldwide, in the US, are infected with HPV. And about 5% of all pap smears contain HPV infected cells. About 10% of women are going to be infected with what's known as high-risk HPV types. High-risk HPV types are more likely to develop into cervical dysplasia and ultimately cervical cancer. So this is a really prevalent infection that has some really severe consequences. So besides cancer, what else does HPV cause? So this is just kind of a list of them. So there's common warts, which are basically what's shown here. Common warts, sometimes also known as plantar or flat warts. These are those papillomas, those condylomas. They're common in children, young adults. It's basically a benign, self-limited proliferation of the skin. It's going to regress most of the time on its own. Most people with HPV infection have a common type of the virus, which is largely HPVs 1 through 4, which basically these are going to infect the keratinized surfaces, predominantly on the hands and feet, as I'm showing here, this person's finger. And the incubation period before a wart develops can be as long as 3 to 4 months. And then the appearance of the wart can be either flat, which is largely what's being shown here, or it can even be a little domed with the wart kind of on top, okay? Or plantar. It kind of depends on the HPV type at the infected site. Innate immunity and cell-mediated immunity are both important to control and resolve the HPV infection, but most of the time they are self-limited. If not, there are various treatments that we'll talk about in a second. So let's talk about some of the other things. Head and neck tumors. Single oral papillomas are the most benign epithelial tumors of the oral cavity. You can also see laryngeal papillomas. Those are associated with HPV 6 through 11, and they're the most common benign epithelial tumors of the larynx. As many as 80% of the oropharyngeal carcinomas contain high-risk HPV DNA. And again, you're getting these head and neck tumors through close contact, sexually transmitted infection of that manner. Okay. There's also kind of just HPV of the sexually transmitted kind. A lot of HPV infections are asymptomatic. We're not going to see anything by it. It's going to show up in a woman's abnormal pap smear. That's pretty much how we find out. But HPV can also be the cause of anogenital warts, which is pretty much what I'm showing here. These are various forms of anogenital warts. This one is actually oral papillomas, more associated with the laryngeal ones, it looks like. But yeah, you might see those. Skin or benign warts are sometimes known as verruca vulgaris, and these are specific for the cutaneous epithelial surfaces. You might also see the term on the anogenital cavity, condyloma acuminatum. Most of these, again, are caused by HPV 6 and 11. You can also see anal and penile warts, which can progress to cancer if they're caused by a high-risk oncogenic strain of HPV, and kind of an uncontrolled infection in patients with the genetic disorder epidermidoclasia verruciformis. Those patients are at a higher risk. Remember, we're going to talk about the high-risk types more on this in a minute, but largely the two you need to remember are 16 and 18. These are the high-risk types that are associated with development of cervical cancer. All right, let's talk cervical cancer here for a second. In a normal setting, we talked about this a lot at the beginning of this video, the virus makes its way down to the basal layer, infects, and then matures on its way up, and then eventually will get some virion production. This is in a normal, susceptible, permissible cell. When that doesn't happen, when you have a non-permissible cell, that's when you can wind up with this invasive cancer, because what happens is the HPV DNA integrates into the host cell genome, and then you don't actually get virion production, but you do get changes to the cell. Like I said before, HPV is normally asymptomatic, and the only way we can detect it really is by finding koilocytotic cells. And that is actually detected by pap smears. We can probably ask Dr. Bradrick how to pronounce this without me butchering it, and that'll be more interesting. But anyway, so you can stain them on a cervical smear. The pap smear is used to detect also cervical carcinoma. What happens is that you take a scraping and you do, this staining, and what you might see is some mild dysplasia, or basically mildly abnormal cells. About 40 to 70% of the time, this mild dysplasia is going to regress. So just because you had an abnormal pap doesn't mean you're going to get cancer. However, at that point, they're probably going to want to do some PCR or something to determine, is it a high-risk type of HPV or not? Types 16 and 18 are the most prevalent of the high-risk types. But there are commercial assays available that enable us to detect 12 other types that may lead to cervical cancer. Okay, so what leads to HPV causing cervical cancer? It actually has to do with this viral protein, E2. So what happens is when the virus integrates itself into the host genome, E2, which normally works to cause the cell to cycle, kind of inactivates the growth stimulation. And neoplasia high-risk HPV types, namely 16 and 18, are high risk because they can initiate the development of cervical carcinoma, as well as oropharyngeal, esophageal, penile, and anal cancers. Almost all the cervical carcinomas contain integrated HPV DNA, with 70% of them coming from HPV 16 or 18. So what happens is you need to break the circular genome with the E1 or E2 genes to promote integration. And that causes these genes to become inactivated. So we lose E1 and E2, but the genome is now integrated into the host genome. This inhibits viral replication, but it does not prevent certain viral genes from being expressed, namely E5, 6, and 7. The E5, E6, and E7 proteins in HPV 16 and 18 have actually been shown to be what is kind of oncogenic. These are the oncogenes which promote the invasive growth of abnormal cancerous cells. To diagnose it, first you're just going to look at the warts and you're going to confirm them microscopically. You can do this via finding prickle cells, hyperkeratosis, obviously an abnormal pap, things of that nature. There's really no cell culture system for this one. We can't grow the virus out. There's a hybrid capture assay. It's known as DiGene. And this is how we can identify 13 different high-risk types. But not individual types, just whether or not the patient has these high-risk types. There's also a linear array, and that uses DNA hybridization, again, so that we can identify types of HPV that might be present. And then we can do real-time PCR. This is mainly used to detect HPV 16 and 18, but there is also this available for 12 other high-risk types. Again, they'll be detected but not individually identified. How do we prevent it? Well, first, you could just avoid close contact. That's an option. For, warts on the hands and feet, you can use cryotherapy. You can also use surgical removal as an option, but our best bet, a vaccine. We have vaccines. And this is a vaccine that's come under fire a lot. And I think part of that is that it's new and that there's still a lot of epidemiologic data that needs to be collected, but it is effective. So Gardasil, there's a quadrivalent one, which basically protects against 4 of the types. So 6, 11, 16, and 18. And if you remember from previous slides, that's going to get you pretty far. 6 and 11 is largely involved in some of the mucosal types, whereas 16 and 18 is going to be more protective against cervical cancer. And then there's nonavalent, and that's going to protect against 9 different ones. So we still have the original 4, 6, 11, 16, and 18. But then in addition to those, it protects against 31, 33, 45, 52, and 58, which means you're getting a lot of protection against HPV, basically.